Hello, guys. Good morning, or good afternoon, or good evening, everyone. Thank you all for joining today's class on antigen antibody interactions. And I am Miss Merle, and I'll be guiding you through this fascinating topic, I hope. And just to share a bit about my background, I hold a master's degree in immunology, and I'm excited to dive into the specifics of how antigens and antibodies interact within the immune system, and we'll cover how these molecules recognize each other and trigger immune responses, which are essential concepts in immunology. And please feel free to ask questions or share your thoughts as we go along. So let's get started, guys. For the learning objectives on precipitation, so after finishing this lecture, the students will be able to first describe how the law of mass action relates to antigen antibody binding, and second, distinguish precipitation and agglutination, or differentiate precipitation from that of agglutination. Third, discuss affinity and avidity and their influence on antigen antibody reactions. Fourth would be to explain how the zone of equivalence is related to the lattice of hypothesis and differentiate between turbidity and nephelometry and discuss the role of each in the measurement of precipitation reactions. But we can omit number five. And sixth, explain the difference between single diffusion and double diffusion, followed by give the principle of the endpoint method of radial immunodiffusion. Eight, determine the relationship between two antigens by looking at the pattern of precipitation resulting from Ouchterlony immunodiffusion. Nine, describe how rocket immunoelectrophoresis differs from radial immunodiffusion. And 10, compare immunoelectrophoresis and immunofixation electrophoresis regarding the placement of reagents, time to obtain results, and limitations of each method. And for the learning objectives on agglutination, the student should be able to differentiate between agglutination and precipitation, discuss how IgG or IgM differ in their ability to participate in agglutination reactions, describe physiological conditions that can be altered to enhance agglutination, and fourth, describe and give an example of each of the following, because these are the types of agglutination. We have direct, passive or reverse passive agglutination, agglutination inhibition, hemagglutination inhibition, and co-agglutination. And fifth, explain and give an application for the direct Coombs test, discuss reasons for the use of indirect Coombs test, and discuss conditions that must be met for optimal results in agglutination testing. So, guys, always the combination of a specific antigen with a specific antibody plays a very important role in the laboratory diagnosis, mainly in diagnosing many different diseases. And hence, immunoassays have been developed to really detect either antigen or antibody, and they vary from easily performed manual tests to highly complex automated assays. But largely, the things that we're doing in the lab here in the university, we're doing more on the manual and qualitative types of tests. And many assays are based on the principles of precipitation or agglutination, guys. So next, precipitation, agglutination, and complement activation, guys, are sorry, immune system processes that are involved in defending the body against pathogens. So let's start with precipitation. It happens when soluble antigens, examples like toxins, and they bind to antibodies, forming a visible insoluble complex. So, soluble antigens, soluble antibodies, and then the result will always be an insoluble precipitate or an insoluble complex, making it easier for the immune system to remove those harmful substances that are present in the precipitate. And agglutination involves the clumping of cells like bacteria when antibodies bind to antigens on their surface. And this clumping really helps phagocytes to efficiently engulf and destroy multiple pathogens at once. And we also have complement activation, and it refers to the activation of the complement system. So, what is the complement system? So, it's a series of proteins that enhance immune responses. It can lead to the destruction of pathogens by promoting inflammation, aiding in phagocytosis, or directly lysing microbial cells through the formation of MAC, or the membrane attack complex. And these processes, guys, work together to neutralize and eliminate threats from the body. So that's with the key points for types of interactions that are involved in our antigen antibody interactions. And next we have what's the difference between a multivalent and that of a univalent antigen. So monovalent and univalent are terms that are often used interchangeably to describe antigens with a single site of interaction, but they originate from different contexts. So monovalent antigen, so in its immunological context, it refers to an antigen that has a single epitope or a single antigenic determinant capable of binding. It's capable of binding to a specific antibody, meaning monovalent means the antigen has only one binding site for antibodies, making it capable of forming only one antigen antibody complex at a time. And then our univalent antigens, meaning in an immunological context, univalent could be used to describe an antigen with a single reactive group, but this term is less frequently used than monovalent. So that's it for our multivalent and univalent antigens. So we also have how can we differentiate Fab fragments from Fab2 fragments? So both are antibody fragments obtained by the enzymatic digestion of immunoglobulins, but they differ in structure and function. And so here are the key differences. So when it comes to the structure, Fab fragment antigen binding. So fragment antigen binding. They're composed of one light chain and part of one heavy chain. And it contains a single antigen binding site produced by papain digestion of an antibody, and each fragment is monovalent. What do you mean by monovalent? It can bind to one antigenic epitope only. How about Fab2 fragments? So composed of two Fab fragments. And these fragments are connected by a hinge region. So by disulfide bonds, contains two antigen binding sites and produced by pepsin. It's produced by pepsin digestion of an antibody, and it is divalent. It can bind to two antigenic epitopes simultaneously. And with respect to valency, Fab fragments are monovalent, meaning can bind to only one antigen epitope. Well, for the Fab fragment, a Fab2 fragment is divalent. It can bind to two antigenic epitopes, increasing its potential for cross-linking antigens. And in consideration with the size, of course, the fragment is smaller due to the cleavage of one antigen binding arm, while the Fab2 fragment is larger because it contains both antigen binding arms and the hinge region. And considering the production of these fragments, Fab is generated by the digestion with papain. Okay, so this one for pepsin, and this one was generated by the using papain digestion, which because papain cleaves above the hinge region, producing two Fab fragments, while Fab2 fragments they are generated by the digestion with pepsin. which cleaves below the hinge region, leaving the two Fab fragments connected. Okay, and also, guys, when it comes to their functions, so a Fab can bind to antigens but lacks the ability to cross-link or form large antigen antibody complexes. But Fab2 can still, it's able to cross-link antigens due to its valency. It's bivalent, which enhances its ability to form larger complexes and potentially elicit stronger immune responses. So, in short, Fab fragments are smaller, they are monovalent, and they are produced by papain digestion. Sorry, guys, it has to be papain here, sorry. And then for while Fab2 fragments are larger, they are divalent and produced by pepsin with enhanced cross-linking ability due to their two antigen binding sites. So this one is papain and this one is pepsin. Please erase this one. So next, so IgG, the most abundant type of antibody in the immune system, can be enzymatically cleaved by papain and pepsin to produce different fragments. So here, to be clear, so for Fab and Fab2, as well as the FC region, okay? And so, guys, how does this process work? So, cleavage by, sorry guys, the cleavage by papain is a proteolytic enzyme. Papain is a proteolytic enzyme and it cuts the IgG molecule above the hinge region. When IgG is cleaved by papain, it generates or it will give you two Fab fragments. Each Fab fragment contains a single it has a single antigen binding site and it consists of a variable and constant region. It is the variable and the constant regions of one heavy chain. And one FC or fragment crystallizable fragment. This fragment includes the lower portion of the how can I say the lower portion of the two heavy chains, including the hinge region here. So the FC region is sorry important for binding to FC receptors on immune cells and activating the complement. So it's more on effector functions. And then next, we have the cleavage by pepsin. So pepsin, another proteolytic enzyme, cuts below the hinge region, closer to the FC part. So that's why it looks like this. So when IgG is cleaved by pepsin, it generates one Fab2 fragment, one Fab2 fragment still connected to the hinge region. So the Fab2 retains the ability to bind two antigen molecules, mimicking the intact antibody's bivalent binding capacity. And the FC region fragments, unlike papain, pepsin degrades the FC. So you'll the digestion using pepsin, you cannot observe an FC portion being produced. So the since the FC region is degraded by pepsin into what miss into smaller peptides, so it is not functional after cleavage, so it's no longer there. So if you'll ask me, FC is produced with what type of cleavage? Only in papain, guys. So next, here, so we have immune complexes. So immune complexes, antigen antibody complexes. So a combination forming a complex. So cross-link immune complexes help in they really function in neutralizing, destroying, and removing microbes or pathogens, and they activate the classical complement pathway to lead. to the process, it leads to the process of phagocytosis, so cell engulfment and particle digestion. So high density FC regions and immune complexes, they really accentuate binding, they enhance, they increase binding to phagocytes. Hence, phagocytosis allows the removal of antigens. And guys, key points for the role of complement, especially the C3 binding. So the complement composed of the classical, the lectin, and the alternative pathways. It is a crucial part of the innate immune response, and it consists of a series of proteins, no, that work together to defend the body against pathogens. And it has three main roles, the complement system. So it has opsonization. So in what happens during opsonization means, so with opsonization, guys, complement proteins coat the surface of pathogens, making them easier for immune cells to recognize and engulf through phagocytosis. So that's for the first one, opsonization. Second is cell lysis or lysis. So once the complement is activated, there is activation. It leads to the formation of the membrane attack complex, the MAC here. And this it makes pores or holes in the pathogen's membrane, causing cell lysis and death. That's why there's cell lysis, because of the membrane attack complex that created the holes or pores in the cell membrane. And the third role of complement is inflammation. So complement proteins, they really promote inflammation by recruiting immune cells to the site of infection, enhancing the overall immune response. So overall, guys, the complement, all these three, amplifies the effectiveness of antibodies and phagocytic cells in clearing infections. So that's it with the complement. So, what's the difference between precipitation and agglutination? So, if we're talking about soluble antigens binding to soluble antibodies and the products are insoluble complexes, that's precipitation. Hence, we can hear the word precipitates, and discovered by Cross in 1897. How about agglutination? Agglutination is really more on particulate antigens and binding with antibodies, and hence we can hear the word agglutinates. Okay, and also precipitation and agglutination when it comes to their mechanisms here. Precipitation really requires that the antigen be really multivalent, meaning it has a lot of epitopes, and that the antibody or antibodies are capable of cross-linking these antigens. So when the antigen and antibody interact, they form that very stable structure, that lattice structure that becomes insoluble and precipitates. How about agglutination? Somehow it's similar to precipitation, guys, but it needs that the antigen be particulate and multivalent. So somehow they are having similarities. And the antibodies cross-link the particles, causing them to clump together. So it's more on particulate clumping. And just don't use the word particulate, sorry, agglutinate. And then, guys, so for precipitation reactions in immunology, so they refer to the interaction between a soluble antigen again and its specific antibody, leading to the formation of that insoluble complex, soluble antigen, soluble antibody, but the end point is this insoluble complex. So when the antigen and antibody are present in optimal proportions, they form visible aggregates or precipitates in solution. So, and largely, precipitation reactions are used to determine the presence of antigens or antibodies in test samples. So, guys, precipitation. So, here we need to remember these two very vital terminologies. So, we have affinity and avidity, because both are key factors that influence precipitation reactions. So, affinity refers to the strength of the binding between a single antigen binding site of an antibody and a specific antigen. So, this is the initial force of attachment, or yeah. So, high affinity antibodies form more stable antigen antibody complexes, which enhance the likelihood of forming visible precipitates in a reaction. And then how about avidity, miss? Avidity is the summation. This is the overall binding strength of multiple antigen antibody interactions. So again, it describes the overall binding strength when multiple antigen binding sites in an antibody interact with multiple antigen molecules. Even if individual interactions are weak or low affinity, but when combined with high avidity, so the interactions can be strong enough to produce a stable complex. So there is compensation, guys. So higher avidity can increase the efficiency of precipitation. So here, theoretical consideration. So the law of mass action. So in precipitation reactions, both the law of mass action and the principle of lattice formation play crucial roles in the formation of antigen antibody complexes. So the law of mass action, this law governs the interaction between antigens and antibodies, stating that the rate at which these molecules bind is proportional to their concentration. So a higher concentration of antigen and antibody increase the likelihood of binding, so leading to a greater formation of immune complexes. And conversely, if one component is in excess, then the reaction shifts to favor dissociation, so potentially reducing the effectiveness of precipitation. So, on the other hand, the principle of lattice formation, this involves the cross-linking of antigens and antibodies into a large, stable network. So, for a visible precipitate to form, multiple binding sites on antibodies must interact with multiple antigen molecules, so forming or creating a lattice like structure. So, optimal proportions of antigen and antibody are necessary for this lattice to form effectively. So, if there's too much or too little antigen, lattice formation is incomplete and precipitation may not happen. So, altogether, these principles determine the efficiency and visibility of precipitation reactions by influencing the balance and structure of antigen antibody interactions, guys. And so, affinity again. So, in precipitation reactions, guys, affinity refers to the strength of the binding between a single antigen binding site of an antibody and its specific antigen. So this is the representation, it represents the likelihood that the antibody will bind to the antigen based on the complementarity of their shapes, charges, and molecular interactions. So, for the types of non-covalent bonds involved in precipitation reactions, so what are non-covalent bonds? So, these bonds are weak, there is reversible interactions that play a crucial role in stabilizing antigen antibody complexes and other biological structures. So, the main types include we have ionic bonds, hydrogen bonds, hydrophobic bonds, and van der Waals forces. So, for the ionic bonds or electrostatic interactions, these are also known as electrostatic bonds. So these happen or occur between oppositely charged groups on molecules, such as the positive charge of a basic amino acid and a negative charge of an acidic amino acid, guys. So they're relatively strong, but weaken in the presence of water or high salt concentrations. However, for hydrogen bonds, these bonds form when a hydrogen atom is what shared between an electronegative atom, like oxygen. See, or nitrogen. So here we have oxygen as the example, hydrogen and oxygen, and another electronegative atom. So they are essential in stabilizing secondary structures like alpha helices and beta sheets in proteins. It's more hydrogen bonds are really important in proteins. And hydrophobic interactions, these happen or occur when nonpolar molecules or regions of molecules aggregate to avoid contact with water. So hydrophobic interactions drive the folding of proteins. So in their presence, proteins fold, and the binding of antigens to antibodies in aqueous environments. So largely likened to our samples because our blood is water based. And then van der Waals forces. So the interactions between electron clouds of oscillating dipoles. So these forces are weak forces, short range forces that arise from transient dipoles in atoms or molecules. So when molecules are close enough, these dipoles induce what complementary dipoles in neighboring molecules, leading to their attraction. So that's it for the non-covalent bonds, guys. So let's shift to the characteristics of non-covalent bonds. So these bonds, again, they are weak and they can only act over short distances. So the Stevens is really specific. So this is just the distance, 1 by 10 to the negative 7 mm. And non-covalent bonds really need a very close fit between an antigen and antibody for effective binding. Sorry, guys, the chair is like farting. Sorry for that. So here, the strength of attraction, guys, is largely dependent on the specificity, cross-reactivity, and optimal binding. So specificity is largely dependent on the binding of an antibody to a particular antigen as governed by the shape and fit of the antigenic determinant or the epitope, while cross-reactivity is on the structural homology of antigens, yet still it is dependent. It depends on the point how close a cross-reacting antigen resembles or the original antigen. And optimal binding, so this one. So if there is this perfect lock and key fit, then maximum affinity happens. So avidity in antigen antibody interaction. So let's define avidity. So it refers to the overall strength of binding between a multivalent antibody and a multivalent antigen. So unlike affinity, which describes the strength of a single antigen antibody bond, avidity considers it is really the reflection of all the cumulative effect of all the binding, guys. And the function is this one, the sum of all the attractive forces between antigen and antibody. So it's the measurement of the overall stability of an antigen antibody complex. That's avidity in contrast to affinity. So avidity compensation. So a high avidity can really compensate for low affinity to achieve a stable antigen antibody interaction. And the overall stability is increased in the presence of high avidity. And largely, there is a good avidity if the reaction involves multiple binding sites on both not only the antibody but as well that of the antigen. So the law on mass action. So the definition it governs the reversible nature of antigen antibody binding and the equilibrium between free and bound reactants. So the equilibrium constant in the context of the law of mass action, this represents the ratio of the concentrations of the products to the concentrations of the reactants. Okay? When the reaction has reached equilibrium. So for antigen antibody interactions, this equilibrium constant is, how can I describe that? It reflects the balance between the formation and the time that there is this dissociation of antigen antibody complexes. So in short, guys, a higher Ka or equilibrium constant is an indication. It indicates a stronger binding affinity, meaning the antigen and antibody tend to stay bound. So it favors the formation of a complex, and a lower equilibrium constant, it suggests a weaker interaction or suggests weaker interactions with more antigens and antibody remaining in their free states. So to conclude, the equilibrium constant measures the stability of antigen antibody complexes with higher values indicating a greater tendency for binding. So the higher your equilibrium constant, so it is an indicator that there's a greater tendency for binding. So which is really essential in predicting the effectiveness of immune reactions like precipitation. So that's it. And then the law of mass action. So the goodness of fit, guys, was the significance of the law on mass action. So the equilibrium constant, it represents the strength of binding between an antigen and an antibody, while avidity impact, it talks about higher avidity decreases the likelihood of complex dissociation, resulting in a higher equilibrium constant value. So that's with avidity impact. How about sensitivity? So a high equilibrium constant, it indicates more antigen antibody complexes, making the reaction easier to detect. So we're referring to sensitivity, meaning even the smallest amount of the analyte can be detected. And for the ideal conditions, so high affinity or initial attraction. Affinity is the initial attraction, remember, and avidity is the summation of all the binding forces. So high affinity and high avidity, what guys? What's the next thing? So again, high affinity, initial attraction, high avidity, overall binding strength. They enhance the sensitivity and detectability of the tests, the ideal conditions. So we really need to follow the proper antigen antibody binding ratio. So here, the precipitation curve, this is used to really describe the precipitation reactions in a graphic representation that happens when the concentration of one reactant is constant. So it's just constant, while the other one is increasing. So for example, guys, yeah. So while the second reagent's concentration is serially increased in the test samples. So for example, here we have an increasing amount of your, what can you notice the difference? Increasing amount of antigens and a constant amount of your antibody. So this reflects that precipitation increases with soluble antigen concentration up to the zone of equivalence. So we only have an equal amount of antibodies corresponding to their specific antigens in the zone of equivalence. And beyond this point, there is already this excess of, so zone of antigen excess. So this is the post zone. This is the known as the prozone. So zone of antibody excess. Excess of antibodies, see? Excess of antigens. Zone of antigen excess, post zone. Prozone, zone of antibody excess. So here, a closer look. So prozone, this is the early part of the curve. So going back, here, early part of the curve, and it shows the presence of free or excess of antibodies or antibody access. Here, the antigen combines with only one to two antibody molecules, and only one site, an antibody molecule is being used, and large immune complexes can be formed due to antigen insufficiency because we've got too many antibodies. So Here, antigens are insufficient in number. That's Burrow zone. And now we are in the middle part of the curve, the zone of equivalence. So lies in between the early and the latter part of the curve. So in the solution, there are no free antibodies and no more free antigen molecules. See? So there is this maximum, optimum amount of precipitation happening here. And the number of your multivalent sites of antigen antibody is approximately equal. So that's the zone of equivalence, forming large complexes of antigens and antibodies. So this the latter part of the curve here. So the post zone. So in contrast, free antigens are present in the solution. So the zone of antigen excess. So there's a gradual decrease in the amount of precipitation, of course, until no more precipitation can be seen or observed. And every available antibody site is bound to a single antigen, and no cross links are formed, guys. So here, immunodiffusion. So there is diffusion based on antigen antibody interaction. So diffusion, the type of diffusion that is, it is the term used to describe the diffusion of antibodies and antigens. So typically, usually immunodiffusion is done or performed by utilizing agarose gels. So specifically 0.3 to 1.5% or agar. This is the supporting medium or support medium because this gives a very stable matrix against temperature and currents. And well, the agarose gels can still permit an unimpeded flow of proteins because the advantage of using agar or agarose, it makes the precipitin bands visible. See, these are the precipitin bands, and the diffusion process is really stable by using agar or agarose. So what's the difference between the two? So if we got the agar from red algae and seaweed, the purified form of this one is the agarose. So agarose is the purified form, the one you are using also in your media prep in bacteriology. And it's composed of agarose and agaropectin for the agar, while the agarose, which is already a modified purified form, is a linear polysaccharide. Hence, it's more expensive and time consuming comes to its production. But for the agar, it is cheaper and less time consuming. So, what's the main goal of immunodiffusion systems? The main goal is for us to be able to detect antigen antibody interactions by using precipitation as the measure of reaction. Okay, so immunodiffusion techniques with single immunodiffusion techniques. So in this process, guys, one reactant, either the antigen, guys, or an antibody incorporated into the agar is, see, it's incorporated, or meaning it is immobilized in the gel or in the agar, while the other reactant is free to the antigen is free to move and interact with it. So to immobilize a reactant in the gel, it must be incorporated into the gel medium while the gel is still in its liquid form. So the free reactant this time, guys, will just diffuse either in a radial, or linear pattern to interact with the immobilized or incorporated reagent in the agar. So that's the immobilized reagent is the antibody, and the antigen is the one that is moving. It's the antigen that diffuses. It's the antigen that meets the antibody. It's being immobilized, impregnated, or incorporated in the agar. So that's the thing. Then, how about single linear immunodiffusion? So, in this technique, one reagent is typically the antibody is immobilized in a gel or fixed in a gel within a glass tube while the antigen moves in one direction. It's just the same, but here we're using the tube. So the antigen is evenly layered on the surface, so forming a thin layer that diffuses downward into the gel. So when the antigen interacts with the antibody in correct proportions, precipitin band forms, so marking the endpoint of the reaction. So what's the endpoint if you will be asked in the exam, what's the endpoint of your single linear immunodiffusion? The formation of a precipitin band. So next we have single radial immunodiffusion. So radial. So this method described is RID or single radial diffusion. So this is used to determine the concentration of an antigen in a sample. So in this method, the antigen is the mobile agent or reagent, while the antibody is fixed in the gel. So always remember that. So a radial precipitin ring forms, and this is an indicator that there is an antigen antibody interaction. So the endpoint is the precipitin ring. No, yes, it's not a precipitin band anymore, but it's a precipitin ring because this is known as single radial immunodiffusion. So we've got two techniques for the single radial immunodiffusion or RID. So the two techniques we have Mancini, so the endpoint method, and where the antigen is allowed to diffuse to completion when the equivalence is reached and there is no further change in the ring diameter. And the second one is Fahey and McKelvey. So the kinetic method, because see, always remember these lines. The measurements are taken before the point of equivalence is reached. This one, we allow diffusion to complete, sorry. But here, even before the equivalence or diffusion is reached, the measurements are taken already beforehand. So that's Fahey and McKelvey, and we have the Mancini for the single radial immunodiffusion techniques. So this one it's a diagram trying to show you how radial immunodiffusion is done. So agar plates are impregnated with antibodies, like this, and then followed by the loading of the antigen in different concentrations in the middle. So it's the antigen that moves, and the one is fixed are the antibodies, and then followed by a period of incubation, and after rings can be observed forming. And then the precipitate ring will eventually be very visible for determination. So here we have the immunodiffusion techniques. So the reagents move through the gel medium under the influence of an applied field or voltage, and then the antigen moves in one direction. So we have the antigen wells here, and then the endpoint will be the precipitin arcs or the rockets. And here the antibodies are in the gel, and the antigens are moved in the gel by why is it that the antigens are moving? By because an electrical current or an electrical field is being applied. And the endpoint will be the rocket-shaped precipitation pattern. So hence also this is also known as rocket immunoelectrophoresis or voltage facilitated single immunodiffusion because it's the voltage, it's the electrical field or electrical current that is used to let the antigens move into the gel. Okay. So other names for rocket immunoelectrophoresis, so many names, guys. Electroimmunoassay, electroimmunodiffusion, L'Oreal technique, voltage facilitated single immunodiffusion. So you might ask me about how it is applied in the clinical setting. So it's on the determine the concentration of a protein in a mixture, detecting abnormal monoclonal antibodies related to lymphocytic diseases like or multiple myeloma, and detecting food allergens. So here, guys, electrophoresis. Can we review what electrophoresis is? So what is the principle? So the principle involves the movement. There's the movement of charged particles like proteins, DNA, or RNA through a gel or medium under the influence of an electric field. So how it works, guys? So it works through a charge-based separation. So the molecules are separated based on their charge, based on their size and shape. So positively charged molecules or particles move toward the negative electrode, and that's known as the where's the negative electrode, guys? This is known as the cathode, while the negatively charged particles move toward the positively charged electrode known as the anode. Okay, so the medium. So usually the medium of our electrophoresis setup often a gel like agarose or polyacrylamide, and this one really provides resistance, causing small molecules to move faster than larger ones. So in an electrical field, so an electric current is applied. Yes. So this one creates a potential difference that drives the migration of the charged molecules through the gel. So separation and analysis, so molecules are separated into bands. So we can observe bands, guys, based on their movement. Or in the case of rocket immunoelectrophoresis, we can observe not as bands but rocket shaped precipitin what precipitates. Sorry. So these bands can be visualized and analyzed to determine their presence, the size and concentration of their specific molecules. So that's for our electrophoresis. So double immunodiffusion technique. So here, why double? Because both the reactants, both the antigen and the antibody, both of them are diffusing. So the antibody diffuses towards the antigen diffuses towards the antibody, forming a precipitate. Hence, both reagents must be added post gel setting. So both reagents are mobile since they are both diffusing. So common sense dictates us that both are mobile in the gel, so neither antibody or antigen is fixed. That's why double immunodiffusion, hence the name. And each reagent is placed in a well and allowed to make a radial diffusion. Okay. And then double immunodiffusion techniques, we have radial boundary intersection. This is the site where the ratio of the antigen and antibody is appropriate. So, like more of saying this is the zone of equivalence. However, what's the disadvantage? So, very time consuming and it's not so sensitive, and it will like eat your 3 to 4 days for you to detect the reactants with low especially for those reactants that are having low concentrations, guys. And classic double immunodiffusion methods include Ouchterlony and simple immunodiffusion. So guys, here, double simple immunodiffusion. So both the antibody and the antigen diffuse through the agarose gel or agar medium until they meet at an equivalence ratio. So here, the antibody and antigen are placed at some distance from one another in parallel wells that have been cut out. So the wells are cut out of agarose or agar that has solidified on a petri dish or a glass slide. So the what's the endpoint? So precipitation of immune complexes. And just similar with what I have told you earlier, time consuming. That's the bad thing about it. So here, Ouchterlony double diffusion. Most board of examiners love to ask questions out from this topic, Ouchterlony double diffusion. So here, earlier we're talking about two reagents diffusing. Now we have three reagents within an agarose or agar medium in a petri dish. So of course, this is more complicated. We're talking about three reagents here. So we have three wells cut out from the gel rather than having two, and then the wells are arranged at the apeces of an imaginary equilateral triangle. So like this. So here, three reagents are diffusing in all directions. So here, we have three basic patterns that can be observed with our Ouchterlony double diffusion. Ouchterlony. So the pattern of identity, full identity, Serological identity, so it forms a continuous line, meaning results when the unknown antigens into the wells are identical and specific for the antibody present in the third well. So one, two, three, we have three wells here. So there is fusion of the lines at the junction to form an arc representing identity. So meaning there's a common epitope that is present between the reagents interacting. How about the pattern of partial identity? And this is very iconic, that spur, continuous line with a spur at one end. So there's fusion of two lines with a spur. This will surely entail partial identity. So again, pattern of identity, pattern partial identity, pattern of identity, pattern of partial identity, and we also have the pattern of non-identity. So the crossing of the lines, the two lines cross completely. So here, a pattern of cross lines means two separate reaction, a different reaction and another separate reaction, indicating, meaning that there's no common epitope that's being shared by the antigens. So here, this is just the same with the previous one slide, previous slide stated on a different way, yet still yields the same significance and meaning. So this one is seen in Stevens. It's just the same, guys. We have the serological identity, non-identity, and partial identity. So here, counter current or counter immunoelectrophoresis, counter current electrophoresis, voltage facilitated double immunodiffusion. So the method described is this one. And in an agarose gel, guys, so the antigen and antibody move due to an applied voltage rather than diffusion. So again, there is an application of an electric field. There's the there's voltage here. So the antigen and antibody are placed in parallel wells and move toward each other under an electric field. So a precipitin band forms when they meet in proper proportions. So this one somehow is rapid as compared to the previous one, which will really eat your time, really time consuming, and largely for SLE, systemic lupus erythematosus, and we also have the extractable nuclear antigens and Haemophilus influenza that can be detected using this method. Okay, so that's the end, guys, and I will have another recording for the part two. Thank you.